For everyone present and for anyone watching the recording. Um, this is Aaron Plex Sulá from the University of Florida, and he's going to present on GIRAF 1.0 for Plant Pests. This is a global innovation risk assessment framework. In today's webinar, Plex will introduce GIRAF 1.0, which quantifies maps and integrates species-specific geographic risk factors, multi-host landscape, abiotic factors, trade networks for agricultural commodities, port accessibility, and international biosecurity policies. We'll learn how GIRAF helps assess plausible scenarios for the introduction, establishment, and spread of invasive pests, Phthorimaea absoluta, Ralstonia solanacearum, tomato brand rugose fruit virus, and potato spindle tuber viroid. Plex Sulá, PhD in pathology from the University of Florida, XMA. He's worked at the forefront of plant health, environmental change, AI, GIS, and network science in the past 5 years. His mission of teaching has always been to develop leaders who transform plant health with innovation, sustainability, and global vision. Plex, we're really excited. Take it away.
All right, thank you very much for this wonderful opportunity that allowed me to share a little bit about my project here at the University of Florida, part of my PhD program. So today we're going to discuss a little bit about this framework, the Global Invasion Risk Assessment Framework, or GIRAF 1.0 for plant pests. And one key question that we are trying to answer is to where to deploy surveillance efforts based on information availability for plant pests around the world. And also the question of where we need to mitigate those risks that are available worldwide. Our outline for today's presentation includes discussing a little bit about the challenges and novelties for pest risk assessment. I'm going to provide you some analysis for real-world cases of invasive plant pests. I'll be describing the components of this framework and how we use this framework to understand the potential distribution of 4 invasive plant pests. In general, this framework targets 3 broad components. The first one is trying to understand the pest introduction potential via international trade networks. The second one is understanding the potential environmental suitability of pest species to understand the potential establishment of those pests. And finally, understanding the potential gradual spread of these pest species across heterogeneous host landscapes. We're going to discuss some potential takeaways for proactive surveillance based on this framework. And finally, I'll be open to any questions, comments, discussions, or monetary contributions. National seed supply often depends on the international movement of planting materials, which may or may not mediate unintentionally the movement of plant pests. An example of this rapid global spread mediated by the tomato seed movement is the tomato brown rugose fruit virus, which reached every continent in less than a decade. Climate change is shifting the conditions that are suitable for plant pests. For example, this paper illustrates how global warming might allow the movement of fungal pathogens towards the poles. But challenges remains for understanding how these environmental changes will affect viral distribution. Natural processes like wind currents also help redistribute plant pests geographically, either at the short distance, for example, the movement of plant pathogens or pests between farms, or at the large distance scale. For example, hurricanes are hypothesized to transport soybean rust inoculum to the United States from South America. There is also this vision of a global surveillance system for crop diseases. And important advances in data management include the development of global databases such as the Gene Bank, the CAVI Compendium, and the European Plant Protection Organization Global Database. Likewise, the International Plant Protection provides an international platform consisting of 185 contracting parties or governments that collaborate to develop, adopt, and promote the application of international standards for phytosanitary measures, or ISPMs. The International Pest Risk Research Group, the IPRRG, is also a dedicated group of research scientists and pest risk practitioners, which aim to develop enhanced pest risk modeling and map methods through rigorous and innovative research. Unfortunately, we lack a global coordinated risk assessment system, which could enhance current biosecurity strategies. Global risk assessments are indeed the exception and often target a few plant pests. In practice, the importance of and the need for pest risk assessments is highlighted by the level of reporting activities by national plant protection organizations. Our proxy in this case is the number of reports an NPPO has regarding national pest management. In general, there are opportunities for filling gaps in pest risk mitigation for many countries, especially for low-income countries. The framework that I am proposing today includes information or data sources related to legislation, entry points, pest reports, and pest status that we can use to improve risk assessment. I know that there are hundreds or perhaps thousands of risk assessments available for a specific country or a few pathogen species or pest species. And here I present five needed innovations for pest risk assessments, which are also contributed by this framework. The first one is that traditionally risk assessments are based on expert opinion. GIRAF offers a new data-driven approach. Most of these risk assessments that are available focus on a national or local extent, while GIRAF provides a multi-scale mapping approach. There are global assessments. Yes, there are. These global assessments often include only generic drivers, but GIRAF incorporates species-specific information on common drivers of pest spread. Most of these assessments that are available focus either on a single risk factor, such as the distribution of cultivated crops or climate variables. GIRAF provides an integrated way of quantifying four different risk factors. And finally, most risk analyses focus on a single often focus on a single cultivated host, but GIRAF is an approach to integrate multiple host species in the landscape. And there are thousands of pathogens affecting plant health, and probably a few hundreds or maybe a couple of them are really invasive. The main objective of our study was to assess the global risk posed by four invasive plant pathogens. The first one is Harringtonia lauricola, which is the main causal agent of laurel wilt of avocado and so many species in the Lauraceae family in the United States and Southeast Asia. The other one is potato spindle tuber viroid, which is associated with the disease in potatoes, such as seed degeneration, but it also can be moved through asymptomatically ornamental plant species. Ralstonia solanacearum is the causal agent of a bacterial blight of potato and tomato, but also can affect some ornamental plants. Today we're going to focus on Ralstonia solanacearum phylotype 2b, sequevar 1. And finally, tomato brown rugose fruit virus, which causes these typical symptoms either on the fruits or on the foliage. And some plant species are also affected by this virus. For example, potato and tomato in Africa has been reported to be affected by this virus, and many other species that are asymptomatically. But why we chose these pathogens? Well, have some traits in common. The first one is that these pathogens have a taste for a wide range of host species. The second one is that these pathogens matter because they cause crop yield losses globally and locally. Global analysis for these pest species are needed because these pests don't respect political boundaries. And finally, They belong to distinct taxonomic groups, making possible to test the generality of our framework. So now let me introduce my GIRAF, which it stands for Global Invasion Risk Assessment Framework. Now let's go to the serious GIRAF that I am proposing. So GIRAF has many specific components, but for now, please let me focus on the four broad components of this framework. The first component is the integration of data sources, either from publicly available datasets or from expert assembled data, as we are presenting in this study. The second is the decision making about which variables are important for understanding the establishment and the spread of these pest species. Next, we build the models and validate these models based on their performance. And finally, we generate maps of invasion risk across scales from global to local. The main goal, in general, for GIRAF is understanding the potential introduction, the establishment, and invasion spread of a pest species. Now let's move to the very specific components of this framework. The first one is understanding the potential international introduction of pest via trade or proximity to ports and cities. This can help us understand where to prioritize international biosecurity at the global scale. To understand introduction risk, we develop a network analysis. So we need to define what is a network. In plain English, a network is a set of nodes that are connected by links. In the case of international networks, we define a network as a set of nodes, where nodes represent countries, and links indicate the possibility of pest movement via international trade of crop commodities. And this can be estimated using different factors. Specifically in this analysis, we ask the following questions to estimate the likelihood of those links. The first one is, how widespread is the pathogen in the importing and the exporting country? How much host is available in the importing and the exporting country? Which traded commodities are associated with the target pathogen? What is the volume of international trade of high-risk commodities? Which countries are currently implementing biosecurity measures targeting these specific pathogens? And how accessibility to ports and cities are associated with pest introduction. Here is the very fancy equation if you are into that, but we can discuss after this presentation about how to include this into your analysis. So here is an example that helps us understand the possible spread pathways for these pests. In this very specific example, the potential movement of tomato brown rugose fruit virus through the international trade of tomato seed. Interestingly, there are 95 countries involved in the trade of tomato seeds, and nearly half is currently invaded by tomato brown rugose virus, making the prevention of introduction of this pathogen into other countries that the virus is not present very challenging. In general, the key takeaway from this type of analysis is that if these historical patterns in international trade continue, there is a possibility that the pathogens move internationally into the tomato producing countries. In the example for tomato brown rugose fruit virus, highlighting proactive surveillance in countries at high risk include the Philippines, Costa Rica, Nigeria, or even the potential reintroduction into the United States. Here is a simpler visualization summarizing the cumulative risk of possible pest introduction into a country from any other different countries in the world. In this case, the first map is illustrating the potential introduction of a Harringtonia lauricola into other countries. Specifically, we included for this analysis the international trade of wood packing material. For the case of potato spindle tuber viroid, we included planting materials, such as the movement of potato tubers and also asymptomatic ornamental species. For Ralstonia solanacearum, phylotype 2b and sequevar 1, we included planting materials, again, seed tubers of a potato and some ornamental species. For tomato brown rugose fruit virus, one of the high-risk factors for this virus is tomato seed, but we also included as a low-risk commodity tomato fruit. Including all this information, the key message here is that there is a large need for proactive surveillance to effectively prevent the introduction of plant pathogens and pests in over 100 countries, where these species are currently distributed. For each pathogen in this study, the United States ranks among the top 10 countries with higher introduction or introduction risk. But each pest species has a very specific geographic pattern for risk that we can use for prioritizing proactive surveillance. For example, for laurel wilt, we have countries such as Mexico and Brazil as important potential introduction points, where we know lauraceae is an important host available in those countries. The next component of this framework is understanding the potential spread at fine resolution based on the availability of hosts and the potential dispersal between those locations, and understand establishment for this pest using host connectivity. In this analysis of host landscape connectivity, all else being equal, host availability is a first approximation to understand pest establishment. For example, in this hypothetical host distribution map, high host density is a proxy for high establishment potential compared to locations where there is not much host. A further step is estimating how easily the pest can move between these locations in the host landscape. And we use network analysis to understand this potential. So we use this information of host distribution and we transform it into a invasion network in which nodes represent the very specific locations in the landscape, and the links between those locations indicate the probability of movement for that species between those locations. In this specific example, we use dispersal kernels based on the distance to estimate the probability of dispersal. Then we summarize back this information into maps to identify specific characteristics of the landscape that are important for surveillance. For example, identifying host hooves, which are those locations that are heavily connected to many other locations in the landscape. And host bridges, which are those locations that are connecting different regions of the network or the landscape, which can be useful for prioritizing the proactive surveillance at the landscape level. And if you are wondering that this type of analysis is complicated, no worries. We have prepared an R package for you, so that is relatively easy to use. So here is a scenario of host landscape connectivity for laurel wilt. Specifically in this analysis, we included over 2,000 plant species that are likely susceptible for these pathogen species. In total, hosts are available in 41 million square kilometers, or approximately 37% of Earth's land surface. And we did the same for the other pest species, where we included not only cultivated plant species, but also invasive and wild plant species around the world. On average, across these different pathogens, between 20 to 40% of Earth's land surface have hosts available for establishment and spread of each pest in this study. Globally, potential host spots for host distribution include Europe and Southeast Asia. In the United States, the information about host distribution is geographically fragmented, as you can see in these maps. But California and Florida appear as important in our analysis. Something important to highlight is that in the case of Africa, the landscape seems fragmented, but a potential limitation is that the sample host in these locations is relatively low, and further improvement we need to develop more this type of framework. Finally, understanding environmental suitability can help us understand pathogen establishment and spread. There is a wide range of potential options that we can use to understand and model the environmental suitability of a pest, depending on the knowledge of a pest species. For example, if we know about environmental associations with the distribution of a pathogen or pest, we can use this information to develop environmental niche models. For example, Ralstonia solanacearum phylotype 2V sequevar 1 infection is known to be affected by temperature, as well as this bacterium is known to disperse via irrigation sources such as rivers or lakes. And so for this specific bacterium, we use this information to develop maps of the potential distribution for this pathogen. On the other hand, there is the scenario where we know very little about which environmental factors are associated with the distribution of these pests. And then, in this case, what we can use is machine learning models trained based on observed geographic outbreaks to understand more or less the potential climate suitability for a pest species. In this case, we used compiled information about the georeference pathogen outbreaks globally, which are the response variables for the models, and 7 climate and soil predictors as the potential explanatory variables in these models. Next, we use 80% of this dataset to train models and 20% to validate these models. We use 4 different machine learning algorithms, including Maxent, Random Forest, XGBoost, and Logistic Regression. We provided maps for each of these models, and we assembled these predictions to provide a global and local maps of potential invasion risk. As you know, pathogens are adapted to very specific environmental requirements. In our study, we were able to predict a large portion of where pathogens are currently reported present. For example, for Harringtonia lauricola or laurel wilt, we use information of the distribution of this species in the United States, and we were able to predict the native distribution of these pathogen and vector species. And the same for the other pest species. For every pathogen in this study, a large area in the United States is estimated to have high environmental suitability when we train the models on the global set of pathogen outbreaks. But also it's important to highlight that some locations where the pathogen has not been reported are also predicted to have a high environmental suitability, so they are important targets to prioritize for proactive surveillance. But also, it's important to highlight that active surveillance should remain in those locations where the pests are currently present. Finally, in this type of analysis for climate suitability, an important question is how climate change will change the distribution of invasion risk. Specifically, in this example, we are illustrating how these climate change scenarios could look like in the future for Harringtonia lauricola. And more or less, they are relatively the same maps, but there are some components that are likely to change at the geographic scale. For example, locations north to the United States and also north part of Southeast Asia and some regions in South America are likely to change and increase in the invasion risks for this pathogen. And this is something that the literature has been highlighted as the latitudinal range shifts for pest species. And one of the components of GIRAF is the ability to provide maps either at the global and local scale. In this study, we focus our local assessments for Florida. And here is an example of how these maps will look like. In this case, we are integrating the three different risk factors into the mapping. For example, in these maps, those pinkish locations are those locations having very high environmental suitability. Those locations that are greenish, such as those in North Florida for potato spindle tuber viroid, are those locations where host connectivity is high. And those locations that are purple or at least whitish are those locations where a high pest trait introduction is likely based on the distribution of croplands close to ports and cities. In terms of validating these models, we only have information about the presence only data for each pest. So we were able to calculate some statistics, but not so much for false positives or true negatives for these studies. For example, for the case of environmental suitability models, we use 20% of your reference presence observations. And in general, there was a good accuracy for each of these models, depending on the model trained, but in general goes from 75% to 92%. Usually Maxen did a relatively better job at predicting the distribution of these pathogens. In the case of host landscape connectivity, we use all your reference observations because that information was not directly included in the models. And in general, between 68% to 87% of those locations where the pathogen is present, our model suggested that those locations were important. And in a statistical test of a Kolmogorov test, we were able to understand that those locations where the pathogen is present usually tend to have a high host lands connectivity compared to those locations that the pathogen has not been reported. For the case of the international trade networks, we validated our model based on a small data set of country-level interception data. Fortunately, we were able to predict the distribution of these interceptions with 100% accuracy, although there was not a high risk for those countries that has been where the pest has been intercepted compared to other countries, indicating that the introduction potential based on trade is very heterogeneous. So the key takeaways from this framework is that GIRAF provides a way of understanding which, how, and where risk factors are key for pest invasions. That at least 22 to 41% of Earth land surface is occupied by host communities for each pathogen. But it's important to highlight that there are some limitations here, and probably this statistic could be higher due to some potential biases in sampling host communities. The United States rank among the top 5 countries with a high trade-mediated introduction potential for each of these pests. And GIRAF could serve as a standard approach for pest risk assessments, whether including other additional variables or not. But the most important message for this type of analysis is that pathogens don't respect political boundaries and travel international without the need of a visa. Some potential future paths for this type of frameworks is that there are some obvious factors that we didn't cover in this framework, including a specific analysis, understanding how time will affect the distribution of invasions, such as seasonal deployment of trade activities or seasonal growing conditions for different hosts. We didn't include international borders, but we are currently working on incorporating these different variables. Another important factor is wind patterns, for example, for vectors or for wind-borne pathogens, or transportation locations at the local level, such as trucks or cars. We need to improve the host distribution maps for some of the host species. We can apply this framework for other pests. If you're interested, I am happy to guide you through this framework, and I am open for collaboration. And finally, there are some knowledge that is not available for publicly available for using this framework. And so integrating expert knowledge is an important avenue for this type of frameworks. So one of the potential limitations for this framework is that information availability is the base. But sometimes there are no much information about the distribution or which hosts are infected by these pathogens. Currently, I am leading this effort of creating a global database compiling epidemiological traits for more than 1,000 pathogen species, information such as the names, the identities, where these pathogens are located, where these pathogens has been reported for the first time, which hosts have been infected by these pathogens at the very specific country level, which temperatures are important or tolerated for these pathogens, what are the dispersal factors, and the environmental variables that matter for each of these pathogens. And so currently we have 1,518 pathogen species in this global database. And if you are willing to collaborate and improve this database, I am happy to tell you more about this one. We are currently in the GIRAF lab, we are currently developing the rapid risk assessment to support mitigation of plant pathogens and pest toolbox, or the R2M plant health toolbox, which includes relatively short components of the GIRAF, but there are also other components that are complementary, such as including expert knowledge elicitation. We have developed the meta question tool that helps us develop these types of analysis, or the scenario analysis for regional management, which can help us understand different scenarios of plant risk. For example, in this paper, we highlighted how different sources of information from published or unpublished information can be integrated to develop different types of modeling approaches. So here's an example where we use the expert knowledge elicitation to provide a risk assessment of the potential movement of with diseases and pests in Pakistan, specifically targeting how these pests are likely to move between different regions of the country via the movement of seed exchange, or characterizing the yield losses at the national scales based on expert estimates. The other one component is understanding the potential distribution of socioeconomic networks in terms of which social actors are driven or managing invasion risk on the invasion network. And so this type of analysis can help us improve our understanding of risk mitigation or proactive surveillance. And with that, I would like to thank everybody that is coming to this webinar, and also my collaborators that helped me develop this framework, but also a special thank you goes to the lab members of the GIRAF lab. I would like to also appreciate the funding from the USDA APHIS and many collaborators that provided their insights and their feedback in developing this framework. A warning is that this work doesn't necessarily represent the views of the USDA and APHIS, it's merely the point of views of the authors of this study. And with that, I would like to thank you everyone, and I am happy to take any questions if we have time.
I'll kick us off with one if you don't mind. I was wondering where you managed to source the host availability data. Is that would have had to be international, right?
Oh, sorry, could you repeat that again?
So, your host availability data. I was just wondering where you managed to source that.
Oh yes, the data sources for those are a couple of databases. So for cultivated species, there is the crop grids database. I mentioned it probably in one of these slides. Let me see if I can come back. The other one is the Global Biodiversity Information Facility. So that one is mostly for the distribution of wild species. There are other databases that we can include depending on the country. For example, for the United States, there are very high resolution maps of different crop species that are not included in those global databases, and there is the opportunity for including that as well. But yes, in essence, I think it was mainly the crop distribution and GBIF for those host distribution maps.
Thank you. Is that to say that you have a question, or are you just raising your hand?
Okay, I had a question. Thank you, Plex, for all of that. Actually, I've got a couple of questions. In the climate change scenario that you put up there, yeah, there was an increase in the suitability. One of the biggest factors there was an increase in suitability in southern China. That looks very much to me like a modeling artifact.
Oh, I see.
Wanted to
Yes,
comment on that.
probably. Yeah, some of the caveats for this slide that I show is that I am only showing one of the scenarios, the global climate models. So this is the potential for variability in those scenarios. So yes, so there is a potential issue with one of the models that we use that we need to take a look on whether the other scenarios or the other global climate models had the same predictions. Yeah, I will say that could be a potential issue.
Okay, because the models that you've used are all correlative models, and
Yes.
Historically they don't perform reliably with climate change. As soon as you go and change the covariates, they tend to fall apart. So I'm just wondering if that seems like an unreliable part to why. So no, I'm struggling here is, yeah, if you're going to use those models, why do climate change if the results are, if you're not going to be confident about them?
Oh, I see. Yes. So the other way that we can also identify whether the model is performing well is using information that is independent to the models. So for example, the temperature tolerance of the pathogen. So for example, looking at those whether they have the same changes and then double checking if those correlative models match with those associated with a more mechanistic way of modeling, right? And we have information for that as well, for specifically for the one that I show was the um, laurel wilt caused by Harringtonia lauricola. We have information of the cold tolerance for those species. And those analyses are also saying the same pattern, that the potential movement towards the poles. But of course, it's not 100% match between the correlative analysis and the more mechanistic models. So, but there is some overlap. Yes.
Right. Yeah, I guess there's a philosophical question there about whether you're better than, for the climate change analysis, going and indicating something that's less precise but more reliable, and just do that, rather, because as soon as you put out fine scale maps, people have this tendency to go and believe them. Okay, okay. Let's see if
Yeah,
there's
I agree. Yeah, there are many different ways for climate change, especially because it's very uncertain, right? And we don't know what exactly will happen in the future. There are definitely another component of these correlative models is that you could use only the environmental space that the model was trained based on the geographic distribution and then using it for the future predictions as well. Because otherwise you will have combinations of the environmental parameters that were not represented in the training data set.
Yeah, using something like the XSTEP tool for that. Okay. That would make it more conservative. Okay. Are there any other questions in line, Jessica?
I don't think so.
Okay, I'm going to ask another question then.
Go ahead.
The data partitioning. So you said that you'd done an 80-20 split. Was that a random split?
That was, so we used two strategies. So one is for cross-validation. So we use key fold. So we divided 80% for cross-validation, and that 80% was divided into different fold numbers. For example, 10 folds for that 80%. And the 20% was used for validating the models. So that data was never seen by the model and was used for comparing to the predictions of the model. So
Right.
it works as a potential independent variable.
Right, but were those that partitioning, was that done on a random selection?
Yes. Yeah, it was on a random basis. Yeah.
Okay, so Town Peterson many years ago did produced a good paper going and showing that, or highlighting that that's not independent data, and it's almost impossible to not get a good result by doing that. That in order to go and test the generality of your model, you need to go and partition that data geographically. You've got to bias it. So train it in one area and then go and see how well the model performs when you test it with data from another area.
Yeah.
Yeah.
So I've seen that approach, the spatial block, I think it's called, or cross-validation, where you have like different, instead of key fold validation, they call it spatial fold validation, where you divide it like the world in different regions. You use one set of that region as the training data and make predictions to the other regions and repeat that process for each of the regions. Yeah, we are also exploring that option of validating the model. Yeah, I agree with that paper that there is not entirely an independent data set if we use only randomly selected observations.
Jessica, Diana's got a hand up.
Yes. Can you hear me?
Yes, yes, we can.
A little louder.
Oh, thank you. Yeah. So in the map where you presented the potential for introduction of the diseases, I noticed that in Central America, like most of them have low potential. And is it because of the lack of register from that area, or why do you think that is?
Well, there are many potential explanations for that. One is that we rely on what is publicly available information in terms of what information about uh, international trade of crop commodities. And one of the potential issues is that the trade activity of those locations has not been reported. The other one is the pest distribution. Although I did an effort of trying to find all the information that was available for each pest distribution, it's very likely that some pests had not been reported officially through scientific journals. And that's a possibility that also need to be updated within these maps. The other one is that the potential distribution of the host, we included the host distribution in these trade networks. And it's very likely that also the host distribution maps for those are not as good as for other high income countries. So those are the potential biases of this framework. Yeah.
Thank you.
Oh, no, Dan's got another question. Never mind.
Sorry, Plex, I didn't catch sort of where is GIRAF hosted at the moment, and how can it be accessed? Or
Yes, so this framework will be available soon after publication. Currently is under review, so we are still developing, but the packages that we are developing are publicly available. For example, the GeoHabnet package is already publicly available in CRAN, so everybody can use it. And we have also the GitHub repository for each of the analyses that we are currently developing. But the framework as a whole, it will be available after this the manuscript will be accepted in the journal.
so
Yes.
so are you expecting in order to use it, people would download a copy and run it locally, or is this going to be an online system that people can log into? Or
Yes, we are pondering still how to deploy this type of tools. So the other framework that I was mentioning is the R2M toolbox, and we are aiming to incorporate all this type of analysis that we develop in this framework in that broader framework, so people can use it either individually or within the ecosystem of the toolbox itself. But yes, currently we are working on that. So for example, we have um, a Shiny app for GeoHabnet, so users can run it online, or they can use the R package in their computers. One of the potential limitations for having it publicly available, for example, the Shiny app, is that if many people want to do it or want to use it, or they want to run a very computationally expensive analysis, and that could be a limitation. So probably it's better to have it as a separate package that they can use it locally. And we are investing on both frameworks. For example, the expert knowledge elicitation, there is also an app that helps develop this type of analysis, specifically targeting or developing the questions. For the trade network analysis, we have an app as well that you will need to only upload the pest distribution and also the information about trade, and the app will automatically generate some types of risk indices for invasion risk. That is still under development. So those three applications are on the way. Yeah. So that's the general aim for this type of analysis in conjunction with the R2M toolbox. Yeah. Soon.
thank you.
I think that might be it for the questions. In which case, thank you very much, Plex. This was absolutely fascinating. Hope to see you hopefully in Honduras in our South American IPRRG meeting next year. And hopefully you will continue to be a part of our IPRRG network, and this will just be really fantastic. Thank you very much.
No, thank you very much for this wonderful opportunity. Yeah, thank you very much for this group. I didn't know until somebody mentioned it, and the dream just come true.
Really happy that you reached out. Yes.
Yeah. Thank you, Aaron. Thanks.
Yeah, thanks. And then we'll also be uploading, I think it was Dimitri who'd asked, just showed up a bit late, so we'll be uploading the whole presentation to our YouTube series as well, so that for anyone who couldn't make the time slot, they can always watch it later in their own time. Means that they can't ask questions, but I'm sure they can always email you if they have any concerns or questions.